Welcome children. In this video I will be dealing with nucleic acid, one of the important biomolecules in the chapter 10 dealing with biomolecules. Nucleic acid, the basic two types of nucleic acids are DNA and RNA. DNA stands for deoxyribonucleic acid, whereas RNA stands for ribonucleic acid. The function of nucleic acid is to serve as the genetic material, but majority of the organism has DNA as the genetic material. Only very few organisms have RNA as a genetic material. We know which are the organisms, certain viruses, and those viruses which has RNA as a genetic material are called the retroviruses. So, besides being a genetic material, usually RNA serves as a catalyst, as an adapter molecule, as well as a messenger. So, these are the usual functions of RNA, as a catalyst, adapter molecule or messenger, but only in few organisms it serves as a genetic material, which is involved in inheritance. Now we can see the basic difference between DNA and RNA. The first and foremost difference is with respect to the number of strands. DNA has double strands, 2 strands, whereas RNA has a single strand. You can see in the picture, this is the DNA with 2 strands and this is an RNA with a single strand. The second difference is with respect to the nitrogenous bases. In DNA, the nitrogenous bases are adenine, thymine, guanine and cytosine. See, adenine, thymine, guanine and cytosine, whereas in RNA the bases are adenine, uracil, guanine and cytosine. So, we can see the difference between DNA and RNA is, instead of thymine, in RNA it is uracil. So, quickly saying about the bases. In DNA the bases are adenine, thymine, guanine and cytosine, whereas in RNA it is adenine, uracil, guanine and cytosine. Third difference is with respect to the sugar. In DNA, the sugar present is the deoxyribose sugar, whereas in RNA, the sugar is the ribose sugar. Now, here warrants a diagram, the chemical structure of sugar. And I am drawing a common diagram for both because this is just the difference of a group that is attached. So, the basic structure is the same for both. So, this is the way you have to draw the diagram. Here we have an oxygen atom, here carbon 1, 2, 3, 4 and 5. Once again I indicate with numbers, first carbon, second carbon, third carbon, fourth carbon and this is the fifth carbon. Now I am going to attach or link or bind the hydroxyl group. Remember, for DNA, hydroxyl group is connected to the first, third and fifth carbon atoms, whereas in the ribose sugar of RNA, the hydroxyl group is connected to 1, 2, 3 and 5th carbon atoms, so excluding 4th carbon atom. So, I am going to draw the ribose sugar of DNA, that is the deoxyribose sugar. So, at 1, at 3 and at 5. So what is this representing? This is representing a deoxyribose sugar. Deoxyribose sugar. Now I am going to make this deoxyribose sugar into a ribose sugar. There is an additional hydroxyl group at the second carbon. Now it has become a ribose sugar. So you got the difference between the deoxyribose sugar of DNA and the ribose sugar of RNA. Once again, you have to draw the structure. I am drawing over this. It should be like this way, like a triangle. And then you have this like this. Then here you have to indicate oxygen atom, then first carbon, second carbon, third carbon, fourth carbon, fifth carbon. Then very important is the placing of the hydroxyl group. If it is deoxyribose sugar, it is at the odd numbers 1, 3 and 5. But if it is a ribose sugar of RNA, an additional hydroxyl group which is at 2 also. And because of this additional hydroxyl group connected at 2, it makes RNA very reactive. One reason why RNA is very reactive is because of the extra hydroxyl group attached to the second carbon of the ribose sugar. Or in terms of DNA, DNA is very opposite of reactive, is very stable. The reason because it lacks the hydroxyl group at the second carbon compound, at the second carbon atom. Clear? So this is the structure, chemical structure of the deoxyribose sugar and ribose sugar. So remember this hydroxyl group in deoxyribose sugar is attached to the first, third and fifth carbon, whereas in ribose sugar the hydroxyl group is connected to, linked to the first carbon atom, second carbon atom, third carbon atom and fifth carbon. So all the carbon atoms except fourth is connected to the hydroxyl group. And definitely you are like science students, you can satisfy the tetravalency of carbon. So here you have carbon, here 1 is over, 2 is over, 3. So 4 with 1 hydrogen. This carbon, how you are satisfying? Here 1 bond satisfied, second bond satisfied, third bond. So the fourth bond with hydrogen. Here 1, 2, 3. So with hydrogen. Here 1, 2, 3. So 1 hydrogen. Here 1, 2 and 2 hydrogens. So that should be satisfied with hydrogen bonds, that you can do it automatically, fill it with hydrogen, single hydrogen or double hydrogen. And the final difference between DNA and RNA is what we said now. That is, DNA is very stable, whereas RNA is unstable or very reactive. And the reasons to support why DNA is stable is because of all the other features. It is double stranded, that's why it is stable. Then the basis T instead of U makes it very stable. And the third reason is the deoxyribose sugar lacks the hydroxyl group at the second carbon atom. So that confers stability. So the fourth feature is very stable. DNA is very stable, and the reason of the stability is all the other 3 features. How you will say the other 3 features? That is, DNA is very stable because it is double stranded. Then it is stable because of T, thymine, makes it very stable. And the sugar it has is deoxyribose sugar, which lacks hydroxyl group at 2. That's why DNA is very stable and usually serves as the genetic material because of the stability. Now talk about RNA, why it is reactive or unstable. One reason, it is single stranded. Second reason is U is very unstable or reactive. If it was T, it would have been very stable. Then the third reason is the sugar. The ribose sugar has an additional hydroxyl group at the second carbon atom, which makes it very reactive. Clear? So these are the basic 4 difference you have to know about DNA and RNA, which serves as the genetic material. Now we are going into the details of the structure of DNA. This is the structure of DNA. first you must remember that I am bringing the 4 bases and I am starting to construct the DNA from its building block. So slowly we will start with the building block. The building blocks are the nitrogenous bases A, T, G and C. A pairs with T, T pairs with A, G pairs with C or C pairs with G. This is complementary base pairing. See, this is a base pair rule, and the bond between the bases is weak hydrogen bond. Between A and T it's a double bond, and between G and C it's a triple hydrogen bond. So you should remember this. Between A and T it is a double bond, and between G and C it is triple bond. A will always pair with T and vice versa, and G will always pair with C and vice versa. No interpairing. Now from the first level I am moving on. So now what I drew is the bases. Now slowly I am going to increase the construction. Here the base is connected to sugar. So I am connecting sugar, sugar, sugar. Now we see this component, base plus sugar forms a nucleoside. So I am introducing the next term, nucleoside. So what is a nucleoside? Nucleoside is sugar plus base. So we see this nucleoside which has adenine and sugar, the second nucleotide which has thymine and sugar, third nucleotide which has guanine and sugar, fourth nucleotide which has, nucleoside which has cytosine with sugar. It's nucleoside. Next I am going to add one more component. The phosphate group. Now what I get is a nucleotide, not a side, nucleotide. So what is a nucleotide? Starting from the left, phosphate plus sugar plus base. So how many nucleotides I have? See, this is the first nucleotide which has adenine as the base, second nucleotide which has thymine as the base, third nucleotide which has guanine as the base, and fourth nucleotide which has cytosine as the base. So now what I drew is nucleotide. Now you can see how many nucleotides are there. You can see 1, 2, 3, 4. 4 nucleotides, and these 4 nucleotides are connected and they form a polynucleotide. So the fourth concept I am introducing is polynucleotide. So what is polynucleotide? It is a polymer of nucleotides. So in this polymer of nucleotides I have 4 nucleotides together repeated, which forms the polynucleotide. So I hope you have understood. From simple to complex, I started with the bases. The bases are A, T, G, C. A will pair with T by a double bond. G will pair with C with a triple bond. So I told about the bases. The bases are A, T, G, C. Next I told about nucleoside. What is a nucleoside? The base with sugar constitute nucleoside. See, base with sugar constitute nucleoside. So accordingly I made 4 nucleosides with the 4 different bases. Then I made it bigger unit which comprises 3 components, that is the base, sugar and phosphate. Base, sugar and phosphate. Now that way, base, sugar and phosphate, 4 of them are made. So we have a chain or polymer of nucleotides. This polymer of nucleotide is known as polynucleotide. So I started from the scratch, that is the building block. Now the same thing I should draw this side. From the base I should slowly construct the nucleoside and then I will construct the nucleotide. Now what I am constructing is nucleoside, that is sugar with the base, sugar with the base. Now I am slowly constructing to the, making it the nucleotide with phosphate. Now I got nucleotide this side also. How many nucleotides? 1, 2, 3, 4. 4 nucleotides. 1, 2, 3, 4. So I got a polymer of nucleotides. So a DNA, if you analyze, a DNA has how many nucleotides? 2 polynucleotides. So DNA has, finally I end up, therefore DNA has how many polynucleotide chains. This is one polynucleotide chain and this is the second polynucleotide chain. So it has two polynucleotide chains. Clear? So once again, the pink indicates one polynucleotide chain which has which is a polymer of a number of nucleotides having the base, sugar and phosphate units repeated. This is one nucleotide, it's repeated, and this side which is indicated in green is the second polynucleotide chains with 4 nucleotides, a polymer of 4 nucleotides. Now, a very important part. Look how it is starting and ending. Look at the start of this chain. This chain starts with sugar and ends with sugar. Look at the other second polynucleotide. Again it starts with sugar and ends with sugar. This is a very wrong representation. So the correct representation is, if this left hand side chain or the first polynucleotide chain starts with sugar, it should end with phosphate. And the second polynucleotide chain will start with phosphate and end with sugar. Now, the ending with sugar will be indicated as 3' end and the ending with phosphate will be indicated as 5' end. So here it is 5', 3'. So we can say the two polynucleotide chains, how are they running? They are not running parallelly. See, parallel means both the arrowhead facing up. This is parallel arrangement. But here the arrangement is like this. This is called anti-parallel arrangement. So here we can say the two polynucleotide chains are running anti-parallelly. The meaning of anti-parallel is here, it can be told in a very brief version. One chain, one polynucleotide chain runs in the 3', 5' direction, while the second polynucleotide chain runs in the 5', 3' direction. This is the better representation of saying anti-parallel. So they are not parallel. Earlier what was depicted is parallel. It both ends in sugar. And this side also ends in sugar, we can say parallel. But here the ending, be careful, adjacent to 3', it should be 5' on the top. And at the bottom, adjacent to the 5', it should be 3'. No 3', 3' together or 5' together of both the strands. Okay, it should be one strand if it's running in the 3', 5' direction, the other strand should be running in the 5', 3' direction. So that arrangement is known as anti-parallel. I have indicated in the form of the structure. Clear?